Ever wonder how your cells know exactly which genes to turn on or off? It's like a symphony of molecular signals, but with a twist. Every note is played in the dark, and the conductor has no score. That's the challenge of gene regulatory networks, GRNs, the hidden code that tells your cells how to behave. Today, we're exploring a breakthrough called PC-GRN, a framework that's rewriting the rules of how we map this cellular software. Let's dive in. So, what's a gene regulatory network? Think of it as the cell's internal traffic system. Genes aren't isolated. They're part of a web of interactions, where proteins act like drivers, signaling molecules like road signs, and regulatory regions like traffic lights. But here's the catch. This system is wildly complex. Traditional models treat it like a fixed blueprint. But in reality, it's more like a city with ever-changing traffic patterns, construction zones, and drivers who sometimes ignore the rules. That's where PC-GRN steps in. This isn't just another tool, it's a whole new way of thinking. At its core, PC-GRN combines three powerful ideas. 1. Category theory. Like a universal language for systems, it helps scientists describe how different parts of a GRN fit together, ensuring consistency even when the details are fuzzy. 2. Bayesian Typed Petri Nets. These are like dynamic flowcharts that model molecular processes with probabilities. Instead of rigid rules, they account for uncertainty, like how a single gene might behave differently in different cells. 3. Generative Bayesian inference. This is the brain of the system. It uses machine learning to guess the most likely network structure and parameters from experimental data, updating its guesses like a detective solving a mystery. Here's where it gets really cool. PC-GRN doesn't just build a single model. It generates a distribution of possible models, showing not just what might be happening, but how certain we can be about it. Imagine if instead of saying, this gene controls that process, the model said, there's an 80% chance this gene is involved, but we're less sure about its exact role. That's the power of uncertainty quantification, turning guesswork into informed probabilities. The framework's secret sauce? A clever combo of two AI tools. GFlowNet, a neural network that explores possible network structures, like a hiker mapping a trail by trying different paths. HyperNetwork, a metamodel that predicts parameter distributions, acting like a translator that deciphers how each part of the network behaves. Together, they create a system that's both flexible and rigorous, able to handle the chaos of biological systems while staying grounded in data. At the core of this mystery are gene regulatory networks, complex webs where genes turn each other on and off, like a relay race. But mapping these networks is like trying to solve a puzzle with missing pieces and shifting patterns. Traditional methods. They're like taking a blurry photo of a city and guessing the traffic flow. They give a single best guess, but biology isn't that simple. There's noise, uncertainty, and hidden rules everywhere. Enter PC-GRN, a game changer that doesn't just guess, it learns. Imagine a team of smart algorithms working like detectives and translators. One part, called a GFlowNet, explores millions of possible gene network structures, narrowing down the most likely ones like a detective eliminating suspects. Another component, the HyperNetwork, acts as a translator, figuring out the exact rules of how genes interact, like decoding a secret language. Together, they create a statistical map of all the most probable networks, showing not just one answer, but a spectrum of possibilities. But here's the real magic. PC-GRN turns these networks into a formal language using category theory, a branch of math that's all about relationships. Think of it like learning how different paths in a maze connect. For example, if gene A influences gene B directly and indirectly through gene C, PC-GRN can mathematically show how those paths combine, like adding vectors in physics. This makes the model not just data-driven, but interpretable, a huge leap for biology, where understanding why things happen matters as much as what happens. Why does this matter? Traditional models struggle with two big problems, uncertainty and scalability. PC-GRN solves both. By learning full probability distributions, it embraces biology's inherent chaos, like a weather forecast that gives chances, not just a single outcome. And by breaking networks into modular building blocks, it tackles complexity without getting lost in the details. It's like building a skyscraper by first mastering the blueprint of each floor, then assembling them. Of course, no breakthrough is without challenges. PC-GRN's blend of advanced math, machine learning, and biology makes it complex to implement. Training it requires solving tricky problems, like keeping algorithms stable or making simulations differentiable, so they can learn from errors. But researchers are already pushing boundaries, testing it on real-world data, refining its tools, and making it accessible to others. So the next time you think about genes, remember, they're not just DNA sequences. They're part of a dynamic, ever-evolving story. And tools like PC-GRN are helping scientists write the next chapter, one network at a time. Want to learn more? Subscribe for more deep dives into AI and biology.